Okay, so again, hello dears, alright? So for this part, okay, so manatagama, kaget mo sa murag flow na to, no, from merely creating the smears lang, like, you know, plain gama ragyo sa microscope, sa slides. And then after, of course, atong butangan ng color, so staining. And then finally, of course, after staining, we examine them under the microscope na. Okay, so this is again microscopic observation of stained smears, okay? So again, this is quite a review na naman of what I've lectured sa Intro to Micro. So we'll again run through lang sa different types of microscope na pod, okay? Alright, so again, microscopy, the use of a microscope to magnify the object not seen by the visible or naked eye. Alright, so itong mga you know, microorganisms, others, basta di dyan makita sa naked eye, di ba? Yung isa itong father of microbiology, you have Antoine van Leeuwenhoek, di ba? Muna ang microscope niya, atong gigamit. And kisa itong scientist na nag-examine sa, nakadiscover sa cells, kaya na-examine siya og cork under the microscope, you have Robert Hooke. Okay. Excuse me. These allergies. Okay. So again, the information on the size, shape, structures, and again, composition, cell arrangement can be seen under the microscope. Again, so nagumantag stain, diba? So our main purpose of staining is to enhance your observation, but we cannot see them under the naked eye. So we need to have the aid of a microscope. Okay. All right. So next, some definition of terms. Again, magnification. You know, the process of visually enlarging objects and the degree of visual enlargement, di ba? Usually, we have 4 types of magnification. 4x for scanner, 10x for low power, 40x for high power, and 100x for oil immersion objective. Now, if ang i-ask kay total magnification, so you multiply this with the magnification of the oculars, which is always at 10x. Okay, so if panguntan mo unsay total magnification, if I'm using the oil immersion objective, then that's 1000x. Okay, usually ang nasa book kana magbasa mo la nag-gi-explain nila, that's total magnification. Kaya mabot ng 400, 100, okay. But ang point again sa total magnification is the ocular magnification. Again, when you say total magnification, it's the ocular magnification, oculars times the objective na magnification. Okay? Alright. Basic. Review na siya. Okay? Alright. Next, you have resolution. When we say resolution, the extent to which the detail is of the image is viewed or is magnified. Okay? And it's dependent on the resolving power. Now, again, sa points of resolving power, the distance lang between 2 points in a picture na pwede pa silang ma-distinguish. So, example, if a resolving power niya is 0.4 millimeters, so therefore, if muna siya ang picture or ang cell, the distance between these two should be 0.4 micrometers para ma-distinguish siya or para makita siya. Okay? Alright. Okay? Understood? So that's the resolving power. Okay. So again, we're na-mention na describe na ako ni sa Intro to Micro. Okay. Alright. Now, we have different types of microscopic methods. First is your bright field microscopy, the most common. You have phase contrast, dark field, and finally you have fluorescence and electron microscopy. So again, these are just some of the microscopes na ginagamit sa lab routinely. Okay? Alright? And sa ito to micro na yung other types of microscope, I'll not go into that na lang. Okay? So kanilang sa. Okay? Alright. So for our first one is bright field microscopy. Again, the most common na itong ginagamit, this is a bright field microscope. Your specimen is usually unstained or stained. So, kano siyang bright field? Because ang iyang background is bright. Okay? So, that's the point, bright field. Resolving power is about 0.2 micrometer. Okay? And half the wavelength usually ang ginagamit ng 0.4 micrometer. And again, ang point objects are viewed against a bright background. Okay? So, that's why it's bright field because ang iyang field or background is bright. And again, the most common type of microscope na itong gigamit. Okay. Usually, tungsten lamp, bacterial cells, are not clearly seen, diba? If wala just like kanang purely ra gyud from broth ba or walay stain, dili gyud siya maklaro kaayo. All right? And here's an example of an unstained specimen. As you can see, wala kay siya color, boring, right? And compared naman pa, yes, after gram staining, you have gram-positive cocci and gram-neg bacilli. So mas ma-identify nimo siya. Compared naman ato, diba? So murag gubot kay siya and then mas naklaro siya under staining. So that's the importance of staining. Okay? All right. Next, we have the phase contrast microscopy. When you say phase contrast, it should be unstained and it passes through a different phases. Now, your different phases, they are converted now to different differences in light intensity or night contrast na mahitabo. And this will help in the identification na pod of, you know, internal structures and different important na mga, you know, structures, yeah. So differences in phase, because of difference in phase, you have differences in light intensity. And what you have to remember lang about phase contrast, it forms a halo of light. Basta gani it forms a halo of light, it's phase contrast. Forms a halo of light in the background of the specimen. Basta halo of light, phase contrast microscopy. Okay, all right. Ayan, so yeah, different phases na enhance contrast and it reveals the gross details of internal structures in a living cells. So here's an example, bright field and then pack. Mas makita na ang internal, okay? So phase contrast, okay? All right. So again, halo of light, na halo sa likod sa mo specimen, di ba? Halo of light. So na siya background na light sa specimen or sa mga structures. Then that's again phase contrast microscopy. Okay. All right. Now for dark field, when you say dark field, opposite sa bright field, actually. So basically, it's just a background, okay? Sa dark. Ang background niya is dark. Therefore, ang imong organism or the specimen or the cell under study, it's bright, okay? Ayan. So specimen is usually unstained and the light is directed at an angle. So walay light na mabot sa objectives. Therefore, wala may light na mabot sa objectives, ngit-ngit imong background, okay? So, and the light will just enter the specimen or the objectives kay na scatter. Reflected light na lang siya, okay? All right. So again, the objects are illuminated. So hayag siya against a dark background, okay? And this is the dark field ring or patch top. Siya ang mag-block sa light, okay? Na para dili siya mo diretso sa objectives. Para produce siya og black or dark na background. Okay. So here's an example. Since this is the dark field ring, right? So dili mo diretso sa objectives. Ma-bend ang light, so outside of the objectives ang masulod. Okay? Only a portion of light, gamay lang. So, dili man tanan, that's why black imuhang background or dark imuhang background. And usually, ginagamit niya siya for your spirochetes. This is an example of Treponema pallidum. Okay? Alright. Causative agent of your syphilis. Okay? Treponema pallidum, subspecies pallidum. Okay, syphilis. Okay? Again, usually ginagamit siya for spirochetes. Ang dark field microscopy. Alright? Okay. Next, we go now to fluorescence. For fluorescence, stained na siya usually. We use certain dyes na ma-excite, o diba? Kung pahayagan sila. And pag pahayag nila, mo release sila, mo emit sila light. Okay, and that's what we see sa microscope. And that's your fluorescence microscopy. So again, objects appear bright. Brightly lit against a dark background. Okay. So again, here's an example, your light source. So of course, ma-excited siya. And then because of a light wave splitting mirror, mato siya sa specimen, na naa dito ang dye, okay? And the dye will absorb the light, okay? And after niya ma-absorb ang light, iyang i-release balik, okay? And that is now the light na itong makita. Okay? Ang light na iyang gina-emit from the fluorochrome, okay? Or the dye na mo absorb sa light. And then iyang i-release balik ang light. And then pag-release niya balik sa light, dito na siya musiga, alright? And dito na makitaan sa imuhang fluorescence microscope, okay? Alright. Okay, so again, we use dyes known as your fluorochromes, and that's your fluorochroming. Some of the dyes that we use are acridine orange, bright orange. Oramine rhodamine is yellow to orange. Chalcofluor white, do not be confused. Ayaw, just because chalcofluor white ihang ngalan, white ihang fluorescence is actually green, blue to white. Or you also have immunofluorescence. Now, unsay difference sa fluorochroming and immunofluorescence? Ang fluorochroming is dye diretso sa cell. Like example, this is the cell, the bacteria. Butangan mo na siya dye dayon. This is the dye. Okay? Alright, that's for fluorochroming. The dye is immediately placed on the cell. Okay? Alright? Whereas for immunofluorescence, you use FITC, fluorescein isothiocyanate, FITC. Okay? Or you also have TRITC, tetramethyl rhodamine isothiocyanate. Ako na nang medyo sayop ko, but we'll focus on FITC na lang. Bright apple green, di ba? Na-mention tamo sa grading sa FITC, di ba? FITC. Ayan. Fluorescein isothiocyanate. Now, ano siyang immunofluorescence? Because, example, this is the cell. Mugamit pa kag antibody. Ayan. These are antibodies. So, ang antibodies mo react sa siya, and then ang antibody na andira ang dye. Okay? So, that's FITC. Mo naman nagtawo, Mark. Okay. Fit C. Okay? So mo react sa daan ang cell og antibody na specific sa cell. Okay? And then, pagbutang nimo sa dye, because of the reaction between the antibody and the cell, mo react pod ang fit C. Okay? That's for immunofluorescence. That's why it's called immunofluorescence. Kaya mo gamit siya antibodies. Okay? And sa itong mga grading sa fit C gani, you have 0, 1, 2, 3, 4. Diba? 0, 1, 2, 3, 4. 0, of course, no. 1 is faint but unequivocal. 2 is apple green, right? Next is bright. And 4 is, 4 plus Denny, brilliant. Okay? Diba? Remember? 0, wala gyud, wala joy fluorescence. 1 plus, naa pero gamay lang. 2 plus is apple green. Okay, like, ana lang. And then 3 plus bright, medyo nagpabibo ka. Bright, 3 plus. And then nagpabibo pa juga intense, 4 plus. Brilliant apple green fluorescence. Please do not take note. Do not take note. Do not forget about the grading of FITSI sa board, sa review na ako ni nahibalaan. So, at least now, kabulo na mo. So, pag-abot sa review, easy na lang. Press the buzzer na. Okay? Alright? Bright apple green fluorescence, 3 plus. Okay. Father of microbiology, Anton van Leeuwenhoek. Cell wall deficient bacteria. Mycoplasma and ureaplasma. Di ba? Medyo nag-loading to. Kinsa to mga gram-positive cocci? Everything except NVM. Okay. And kinsa to mga gram-positive bacilli? Mac Belangaw, BEP. Dapat memorize na guys ha. Muna akong challenge ninyo. Okay. Alright. So for acridine orange staining, usually, according to Bailey and Scott's pod, if usually naka-gram stain po daw na medyo di pa ka-sure, especially from blood cultures daw, mga culture sa blood, para ma-confirm mo siya, you use acridine orange. Okay. Yeah, eliminates the shortcoming of gram staining. But it's non-specific because it intercalates with double helix DNA. Now, most of your bacteria, they have DNA. So it's not that specific. But if the organism has double helix DNA, then na siya bright orange fluorescence. Okay? But it's more sensitive than gram stain. Because again, na-contrast between stained organisms and backgrounds. Since iharang is-stained yun is katong na DNA. And your background doesn't have DNA. So, bahala kung sa kagamay sa organism dito, basta na DNA, ma-detect niya. So, therefore, it's more sensitive than gram stain. Okay? Alright. Ayan. And here's an example of fluorescence microscopy. That's acridine orange. You have oramine, rhodamine, chalcofluor white, and you have FITSI, fluorescein isothiocyanate. As you can see, ang chalcofluor white, as far as I can remember, chalcofluor white, ginagamit niya siya for fungi, mga yeast na mga cell wall, usually. So this is a fungi, as you can see. Fungi na siya. It's too big for a bacteria. Kani, oramine, rhodamine, I think these are MTB, your M tuberculosis na bacilli. Okay? And this one, I think, I don't know, other organism na siya. But again, please take note of the color. Chalcofluor white, do not be fooled by the name. It's color green. Dili siya white. Okay. Alright. And of course, the last type of electron microscopy, by the name itself, it uses electrons, okay, instead of light. And you have two types, diba? You have transmission and scanning. By the name itself, ang transmission, it passes through, so transmit. Therefore, since it passes through the structures, makitan ang internal structures. And for scanning, since it just passes the surface, therefore, ang makitan lang is ang surface rapod, dili ang internal. So if transmission, you want to see the internal structures because again, the electrons transmit or mo pass through the structure. Whereas scanning lang, scan. So ang surface lang. 3D views of the outside, external lang, dili makita ng internal. Because again, the electrons just bounces off the surface. So since mo bounce off the surface ra, ang makitan is ang surface rapod. Okay? So here's an example. Okay, ang left, unsa man na siya? Transmission or scanning? Transmission, because that's makita ng inside. And of course, this one, is it scanning or transmission? So it's just scanning, because makita ang external rana structures. Okay. Alright. And these one, kani mga naa sa gipoint, those are bacilli. I think, and this is a macrophage siguro, yeah, a phagocyte, devouring a bacilli. So these are bacilli, mga gram-negative bacilli. Or yeah, bacteria. Okay, so again, gets lang, transmission, internalization, makita ng sulod. And for scanning, you have just the surface. Okay. Alright, so after nato og examine, of course, we now go to reporting of results. Because we're in bacteriology now, we will report the results of course, what you see. Okay? Now, sa next video na lang. Okay. Okay, so that's all again for microscopy. Now, for the last part in bacterial identification and smear prep, we will now discuss the reporting of results in microscopic exam. Okay? All right.